Dear students, in the previous class we discussed a lot about plant cell structure. There are several components of the plant cell as well as animal cells. In the same class we also discussed prokaryotic cell as well as eukaryotic cells. One thing is common to prokaryotic and eukaryotic cells. They are bounded by one outermost membrane, that is called plasma membrane. Plasma membrane is the structure that is common to both plant, animal and bacterial cells. So, here you can see it is a computer generated plant cell, and plasma membrane, this is very adjacent to the or pressed to the cell wall, not that clearly visible, but if you see this label is plasma membrane. So, along with plasma membrane there are several other membranes. In eukaryotic cell almost 13 different types of membranes are there, because here you can see this is the chloroplast. It has three membranes: outer membrane, inner membrane, and one important is thylakoid membrane. Similarly, this is the mitochondrion called powerhouse of the cell, right? It has two membranes, outer membrane and inner membrane. But we know chloroplast and mitochondria, they are responsible for conversion of energy. This converts light energy to chemical energy, and this converts food energy to ATP energy. Similarly, this vacuole, आमे ओडिया को रसदानी बोली पढ़ी. So, this vacuole or रसदानी that fills most part of the plant cell, parenchymatous or soft cell. In soft cell, this vacuole covers most part of the cell, and this is surrounded by one membrane called tonoplast. So, now what I want to say, this is endoplasmic reticulum, it is membrane. Golgi, it is also membrane. Tonoplast, it is also membrane. Mitochondrial membrane, there are nearly 13 different types of membrane in the cell, including plasma membrane. So, cell membrane is a common term, but plasma membrane is the outermost membrane of any cell, whether it is prokaryotic cell like bacterial cell or eukaryotic cell like plant, animal, fungi or any cell types. Now, but all these membranes, they have different functions, they have different variations in their structure, but thing is they are built in the same basic design. So, we will mostly focus on the plasma membrane. So, plasma membrane is a very thin sheet. Two terms are there, plasma and membrane. Plasma means it is not solid, not liquid, it is of intermediate consistency, thick, olive oil thick consistency of a membrane. So, now it is a thin layer of lipids and proteins that surrounds the content of cell. If we think what gives integrity to a cell, now it is the plasma membrane which gives the confinement to a cell. Without plasma membrane, the cell cannot be confined, it cannot be a compartment. So, cell is a compartment, its outermost boundary is your plasma membrane. Similarly, chloroplast is a compartment, mitochondrion is a compartment, endoplasmic reticulum is a compartment. So, several small compartments are within a big compartment, that is your plasma membrane bound cell. So, here, but in addition to the plasma membrane, beyond the plasma membrane, outer to the plasma membrane, there is cell wall that we have discussed earlier. Now, plasma membrane, the history goes back to 150 years, more than 150 years. You can see some, they coined this term cell membrane, Nageli and Kramer, but the term plasma membrane was coined in 1877 by Pfeffer. Okay. So, the main function of the plasma membrane is compartmentation. Compartmentation, it makes the cell a compartment. Without that, it is not possible. Here I can show you the structure of the plasma membrane. So, plasma membrane is a thin structure. एथिरु आमु केते गुडे जिनिस आमे जानि नाहंती, but we have to understand this. This is an animal cell plasma membrane. Animal cell plasma membrane. This is designed by computer or generated by computer. So, here you can see this structure is a phospholipid molecule. Phospholipid molecule. I told you membrane is lipid and protein. It is made of lipid and protein, but how they are organized, that is important. We have to discuss the organization of the membrane, how structurally it is and how it function. ताने structure सहित structure टा explain करि पारुथो यार function को. तो now this site is extracellular site, because if we think of this is a cell and this is the cytoplasm site and this is the exterior site, सेथि पय एटा लिखा हिची extracellular fluid, ता माने it is outside and this is inside and this is the thickness of the membrane. Okay, here you can mark there are two layers, this is one, this is another, and this structure, a radius head and two black tails, this is a phospholipid, So, now this is one layer, this is another layer. So, this is a bilayer, membrane is a bilayer, and what these structures are? Now, the proteins, these are the proteins, I will be coming to this again, right. So, now, the membrane is a thin sheet of phospholipid bilayer in which proteins are present. Okay. The study of plasma membrane began in 1892. 1892 scientists started reading or experimenting what is the structure, what is the composition, how it is organized in 1892. But almost in 1972 scientists concluded definitively what is the structure of plasma membrane, and that came true. Different models were proposed, we do not have time to discuss, you can go through from the books, but what ultimately membrane टा जेमिति थिबा कथा सेमति अछि, किंतु with the scientist, with the botanist, with the zoologist, with the biochemists आमे ताकू स्टडी करूछंती जे रियली सेल भितरे naturally तार ऑर्गेनाइजेशन टा केमता कोथेरे तयारी आउ केमिति ऑर्गेनाइज हेक अछि. So that took almost 80 years to arrive at a conclusion that this is the fluid mosaic model of plasma membrane. देखो पिले fluid जिनसटा केमती आसे ना liquid आ solid मझे मझे गोटे intermediate consistency रो गोटे जिनस को कहा जाए fluid. तो आमे एठी कहिबा so fluid mosaic model of plasma membrane. When we will come across the subsequent slides, we can understand what is the mosaicism. Okay, so let us go for the features. The fluid mosaic model of plasma membrane was proposed by Singer and Nicholson in 1972, and the important features of this membrane are all biological membranes are chemically lipoprotein in nature. I told you they are made of lipids and proteins. However, then question came, what is the percentage or by weight chemically, what percentage is protein and what percentage is lipids. Okay, but it varies. You can see in the mitochondria has two membranes, outer membrane and inner membrane. Okay, the inner membrane is 76% protein and 24% lipids. एई जिनस 76% प्रोटीन माने सेटा कहूची द मेम्ब्रेन इज हाईली फंक्शनल. आमे देखिबा इनर मेम्ब्रेन ऑफ माइटोकॉन्ड्रियन इज रेस्पोंसिबल फॉर सिंथेसिस ऑफ एटीपी आउ सेथरे इलेक्ट्रॉन ट्रांसपोर्ट चेन चले So, carriers are the proteins present, that is why in that you can find maximum amount of protein, that means it is highly functional. आमे जदि आमे नर्व सेल को देखंदी बा न्यूरॉन को देखंदी, न्यूरॉन चारिपटे के इंसुलेटिंग शीथ थाए, कवरिंग थाए, because one neuron and other neuron they must not interfere. So, neurons are covered by the sheath and they are formed by Schwann cells. Schwann cells सेठी से फंक्शन नेचर नुहे, सेठी से स्ट्रक्चरल रोल करुची, सेथि पई से गोटे कवर बनउची. So, Schwann cells plasma membrane are made of mostly lipids and very few percentage are proteins. Mitochondria inner membrane highly functional, it is most मान 75% or more is protein and 25% is lipids, but Schwann cells which perform structural role that has only 25% or so lipids, proteins, but mostly it is lipids. So, now, but in the plasma membrane generally, plasma membrane has 40% lipids, 40% protein and 20% carbohydrate. Another component came, they are carbohydrate, and carbohydrate in the form of oligosaccharides. Now, so membrane lipids are phospholipids and sterols which form the structural framework of it. Due to hydrophobic interaction between fatty acids, tails, phospholipids and water, lipids organize themselves as a bilayer in water. Now, phospholipid bilayers form the basic structure of all biological membranes. Okay. Now, we can have a drawing on the board, so that we can understand what is the phospholipid structure, right. So, let us have a drawing of that. Lipid is, now phospholipid is a conjugated lipid. आमे जो स्नेहसार पढ़ी ये गोटे टाइप रो स्नेहसार जोटा कि पानी रे soluble usually नुहे, किंतु in this part there is phosphate, in this part there is phosphate PO4 minus. So, this phospholipid and this is called walking stick model. तमे माने जदि कहीं के देखि थिबो जो लोक माने स्टाइलिश walking stick धरंती तार अगिलारे गोटे नॉब टे थाए आ गोटे बाडी टे थाए. So, these are fatty acid tails and this is the head. So, this part is water insoluble, water heating, that is why it is called hydrophobic tail, right. And this part having the glycerol group and negatively charged phosphate, okay, that is hydrophilic or water soluble, So, here I may tell you that this phospholipid molecule is a conjugated lipid, it is not a simple lipid, it is a conjugated. Further, this is amphipathic. आमे गोटे मॉलिक्यूल को कहउछंती जे से होची नेगेटिवली चार्ज्ड, आउ का कहउछंती से पॉजिटिवली चार्ज्ड. जदि जनकर दीटा जाक चार्ज थियो, दैट इज डिफरेंट, ओके, जूटर आयन ता कह जाय. सिमिलरली आमे गोटे मॉलिक्यूल को देखुछंती फॉस्फोलिपिड जार गोटे पार्ट होची, वन पार्ट इज हाइड्रोफिलिक, द अदर पार्ट इज हाइड्रोफोबिक, सॉरी, this part is hydrophobic, water heating, and this part is water loving, okay. So, as a whole this is amphipathic having two different properties, water heating and water loving properties. Now, what is seen that in the membranes, in all the membranes, these phospholipid molecules, they organize themselves as bilayers, Let me tell you little bit what this straight line is and what this bent is. This straight line means this is carbon single bond, and you will have CH2 like this, but here CH double bond CH, wherever you will be finding this bend. This bend means this is a double bond. So, in this fatty acid where there is a bend, that bend is called kink, K-I-N-K. It means it has double bond in its fatty acid tail. So, if it is at all straight chain, only single bonds between carbon carbon hydrocarbon, and if at all there is one or more double bonds, there are two or more kinks. So, now how they are organized in the membrane? The basic properties आमे गोटे जिनस को बुझिबा why they form bilayer lipid bilayer. Membrane is phospholipid bilayer. Membrane is a phospholipid bilayer. Now how they happen, how it happen? So आमे देखिबा सेल बाहरे, एटा सेल रो बाहर, सेल एक्सटेरियर, सेल एक्सटेरियर रे पानी अछि, सेल इंटेरियर रे जोठे पानी अछि, प्लाज्मा में सॉरी साइटोप्लाज्म भी मध्य अछि ए भितरे. ताहले ता माने एपटे वाटर अछि, एपटे मध्य वाटर अछि. कैन दिस एग्जिस्ट as monolayer? No, because this is hydrophilic, this is hydrophilic and this is hydrophobic, hydrophobic, water hating. So this side is water, this side is water. So this is possible. Thermodynamically this hydrophilic can be miscible or in contact with this water present in the exterior, but this hydrophobic tail, in case of it is monolayer, this doesn't possible. That's why to avoid the contact of hydrophobic water hating tails with the cell interior which has water, that's why the phospholipid molecules have organized themselves to form phospholipid bilayer. So now they form phospholipid bilayer like this. Shortly eta kohuche they form a phospholipid bilayer. So this much is hydrophilic, because this is much very much important for understanding the function of this. And this core is hydrophobic. Aku kahanti hydrophobic core. Okay. So this is important. तो पानी या कंट्राक्टर लगी पारो या कंट्राक्टर लगी पारिबो, बट ए हाइड्रोफोबिक कोर रे पानी बा कोनोसी आयन, फॉर एग्जांपल सोडियम वाटर सॉल्युबल, sodium water soluble, chloride water soluble, it cannot pass through from this side to this side through this hydrophobic core. सेथि पई से एपटू एपटू को आसिया पई कोन से आयन से अन्य रास्ता खोजिबे, काहे कि ना ए जो कोरटा होची कोन वाटर हिटिंग आउ आयन मानकू जीवा आसीवा करबा को दबो नाही. किंतु आमे जानि छंती आमे सॉल्ट को खाई दउछंती सेटा जाउची कोठी कि आम गट रे, किंतु सॉल्ट इज अब्जॉर्ब्ड थ्रू द सेल मेम्ब्रेन इनटू द ब्लड. Definitely membrane dei ki sodium, chloride, glucose, all the things they are passing into the cell. Now we will go to the slide. Okay, stop. So basically the lipid bilayer is 5 nanometer thick, but the thickness of the membrane varies from 5 to 10 nanometer due to the presence of proteins and carbohydrates. We will see how they affect the thickness of the membrane. Okay. So now the core of the membrane by opposing fatty acid tails. If you see, these are the two fatty acid tails of this phospholipid and these are two. In this way they oppose each other to form the core as I explained. So the core is very important. Okay. In the fluid condition, the membrane both lipids and proteins move, making the membrane a dynamic structure. What I mean to say, they are flexible. Membrane is not a rigid structure, it is a flexible structure, right. आमे जदि गोटे टब नहीं पानी रो टब से दुई चारिटा पीस डिफरेंट कलर रो बरफ पकई, ओके, विथ पैसेज ऑफ टाइम आमे देखिबा पानी की तो आमे देखि पारु नाहंती पानी मूव करूची ना नहीं, किंतु बरफ आमे जोठी पकईथिबा डेफिनेटली टिके हेले तार पोजीशन शिफ्ट करिथिबो. सो इन वाटर मॉलिक्यूल्स मूव, जोटा कि तमे हळदी गुंडा पकेले सेथरे जानि पारिबो दे मूव. ओके. सो water molecules move and the ice cubes or ice pieces also move, but the movement of water molecules is faster in comparison to the large ice pieces. Similarly, in the lipids, lipid bilayer, the lipid molecules they change their position because of fluidity. Similarly, the proteins. So, lipids and proteins they change their position with respect to time because of the fluidity, Okay. The phospholipids move in their monolayer, but they do not flip flop. मु जो दीटा लेयर करी ऊपर लेयर रु तल लेयर को जमती मांकड़ चित मारती सेमिति हे पर फ्लिप फ्लॉप मीन्स मांकड़ चित मारिबा. सो ए लिपिड मॉलिक्यूल फ्रॉम वन मोनोलेयर or outer monolayer cannot flip to the inner monolayer. It is not possible, but they can move in their own monolayer. So this is because of the fluidity. So now the different functions of the the different functions of the membrane are due to the presence of proteins. In the beginning I told you, inner mitochondrial membrane has 76% protein, means it is highly functional. So all the functions of the membrane are performed by the presence of protein. What lipids do? Now lipids do a structural framework, they give a structural framework. जो सेल जते अधिका काम करजी माने माने कह ट्रांसपोर्ट हो बा एनी अदर फंक्शन हो से निश्चय अधिका प्रोटीन थिबे. So the proteins carry out the different functions of the membrane, okay, but there are two types of membrane proteins depending on their organization. They are extrinsic proteins. Extrinsic means peripheral, they are present on the membrane surface only. For example, मो शार्ट रे गोटे पोकटे बसछि तमे ताकू एंती मारी देले से बाहरी जीवो, जस्ट गोटे सॉफ्टली से अटैच हेतिला किंतु तमे ताकू टिप्पण बा गोटे कोन सेंट्रीफ्यूगल फोर्स देले से बाहर की पड़ोची, गोटे सूताटा को कोन हबो ना तमे छिंडे की बाहर करबो. सेमती द प्रोटीन व्हिच क्रॉस द थिकनेस ऑफ द मेम्ब्रेन that is intrinsic protein or integral protein, जोटा कि मेम्ब्रेन रो इंटीग्रिटी रे थाए, स्ट्रक्चर रे थाए, that is your integral protein. But जे बाहर सरफेस हो बा भीतर सरफेस हो, which is present on the surfaces only, they are extrinsic protein, they are weakly attached to the membrane. Okay. Now if we from this you can see एटा कौन आमे जानी पारुछंदी, एटा मेम्ब्रेन जाइछि, एटा कंप्यूटर जनरेटेड मॉडल मेम्ब्रेन टा केंते कट्टा जाइछि टू शो इट्स ऑर्गेनाइजेशन. सो दिस वन इज द मोनोलेयर, आउटर मोनोलेयर, दिस इज द इनर मोनोलेयर and these are the proteins. This protein crosses the thickness of the membrane. Membrane ର thickness को cross करी एपटू किछि बाहरछि, एपटू को किछि बाहरछि, otherwise membrane टा continuous अछि. So this protein is integral protein or intrinsic protein that spans the entire thickness of the membrane. Okay. So a protein देखि जिंस किछि जीवासिया करे, ये they carry out different functions we will see, right. And this is the membrane. आउ ए मझिरे मझिरे देख कौन कौन गोटे स्ट्रक्चर देखे नाली रे डीप, दे आर कोलेस्टेरॉल्स. आमे पढ़ी कोलेस्टेरॉल बॉडी रे रहिले आर्टेरी गुड़ा सब हार्ड हो जाए, कहते जार जिते कोलेस्टेरॉल डिपोजिट हम तार स्ट्रोक हम. व्हाट डज इट मीन? कोलेस्टेरॉल्स व्हेन प्रेजेंट दे इम्पार्ट हार्डनेस. सिमिलरली द एनिमल सेल प्लाज्मा मेम्ब्रेन टू हैव स्टिफनेस little bit, okay, they have cholesterols in their membrane. So phospholipids and cholesterols they form the structural framework, and the proteins come in two forms. The peripheral proteins, it is just attached to the surface, it can be easily removed, but this integral protein cannot be easily removed. Okay, only when the membrane is broken by detergents, detergent रे जते आपको भांगीबा सेतिकी बेले इंटीग्रल प्रोटीन के बाहर करि परिबा. ओके, अनदर फीचर आम पढ़िले मेम्ब्रेन 40% लिपिड्स, ओके, 40% प्रोटीन्स एंड 20% और सो डिफर्स करे. एगुड़ा होची कार्बोहाइड्रेट, देखो लिखा है कार्बोहाइड्रेट चेन्स. सो कार्बोहाइड्रेट चेन गुड़ाक सदा बेले ये बाहर पटू अच्छी देख दे आर प्रेजेंट टू द आउटर फेस ऑफ द मेम्ब्रेन नॉट टू द इनर फेस। तो आमे जेमिति फ्रंटाली और बैकवर्ड माने आपको कहे वेंट्रल एंड डॉर्सल डॉर्सो वेंट्राली आमे होउछंती एसिमेट्रिक आम वेंट्रल साइड और डॉर्सल साइड आर डिफरेंट। सिमिलरली मेम्ब्रेन रो आउटर फेस इनर फेस दे आर डिफरेंट सेथि पई कहै छी प्लाज्मा मेम्ब्रेन इज एसिमेट्रिक राइट। सो नाउ let us move to the next one what is mentioned intrinsic proteins can be removed by from the membrane by detergent treatment only they act as channels or pumps through which ions and molecules can pass। ये जो जिनसटा जो प्रोटीन माने रहिले से माने कौन हिसाब रे काम करंती ना चैनल देकी चैनल आमे पुणिछंती चैनल गोटे थाए गोटे पोलो भलिया तार गोटे डोर थाए से डोरटा खोली गले पानी ता संगे जहाँ हवा कथा चलिकी पड़े आसो। सिमिलरली आयन्स क्लोराइड दे पास फ्रॉम वन साइड टू द अदर साइड पोटासियम के प्लस मेम्ब्रेन बाहर सेल भीतर को पशिया यही चैनल्स मानकर साहाय्य नियंती आ पंप्स माने पंप जी एनर्जी खर्च करे एनर्जी खर्च करी कौन करे ना मॉलिक्यूल्स मानकू फ्रॉम लो कंसेंट्रेशन रीजन टू हाई कंसेंट्रेशन रीजन को कौन करे ना पंप कर पंप मीन्स अगेंस्ट ग्रेडियंट but the carbohydrates present they are oligosaccharides they are just 4 to 15 monosaccharides long and they are present either attached to the protein forming glycoprotein if they are attached to the lipids they form glycolipids glycos means sugar जेतेबेले oligosaccharides attached to the lipids they form glycolipids when they are attached to the proteins they form glycoproteins and I told you because of the sugars they are asymmetric now the fluid mosaic model has solved the central dogma of membrane biology जे पर्यंत फ्लूइड मोजाइक मॉडल जना पड़ी नथला जे मेम्ब्रेन यही अवस्था रे एग्जिस्ट कर सेल रे से पर्यंत आमे ठीक से जानी पार नथले मेम्ब्रेन केमितिका कारण मेम्ब्रेन थिकनेस होची नानोमीटर लेवल रे नानोमीटर मीटर माइक्रोमीटर देन नानोमीटर मीटर सेंटीमीटर माइक्रोमीटर नानोमीटर 10 to the -9 range ro so it is so thin so it took a long time to study this okay so now the fluid mosaic model of plasma membrane is accepted and कहीं ता कंपेयर करिसेंती जे इट इज सी ऑफ आइसबर्ग्स जेमती समुद्र रे पानी अच्छी से आइसबर्ग्स भासुची सेमती एटा होची सी ऑफ लिपिड्स ए बाइलेयर टाकू लिपिड रो समुद्र कहिसेंती प्रोटीन्स गुडा को आइसबर्ग सहित कंपेयर करिसेंती एंड नाउ व्हाट आर द फंक्शंस carried out by that in the beginning I told you बिना प्लाज्मा मेम्ब्रेन रे सेल एग्जिस्ट करियो नहीं ताने सेल इज अ कंपार्टमेंट it was made possible because of the membrane plasma membrane okay endoplasmic reticulum it is a compartment it was made possible because of that membrane so compartmentation is the basic property of the phospholipid molecules and that made eukaryotic cell our cell a compartment of compartments ता भितर आउ छोटे छोटे कंपार्टमेंट किए ना न्यूक्लियस गोटे कंपार्टमेंट एंडोप्लाज्मिक रेटिकुलम आउ गोटे कंपार्टमेंट राइट क्लोरोप्लास्ट गोटे कंपार्टमेंट माइटोकॉन्ड्रिया कंपार्टमेंट so many compartment lysosome tonoplast they are different compartments small ones integrating among themselves and working within the cell but they are not isolated remember chloroplast is not isolated ये cell cytoplasm रू nutrients नउची एटीपी तैयारी करची कार्बोहाइड्रेट तैयारी करची से कार्बोहाइड्रेट को पुणी कौन करची बाहर को छाड़ुची इन द फॉर्म ऑफ सुक्रोज सो एनी कंपार्टमेंट इज नॉट ए सील्ड बॉक्स इट इज एन ओपन सिस्टम ओपन इन द सेंस से माने न्यूट्रिएंट आउ एनर्जी न्यूट्रिएंट एंड एनर्जी एक्सचेंज करती आउटर एनवायरनमेंट सहित ओके सो द प्लाज्मा मेम्ब्रेन व्हाट आई मीन टू से इट इज सिलेक्टिवली परमेबल आमे कहिबा गोटे गछ जो लगुच से सिलेक्टिवली सब न्यूट्रिएंट्स नौ से गचर जहाँ जहाँ दरकार जेतिकी परिमाण दरकार से नौ हाउ इट इज पॉसिबल बिकॉज of the membrane because of the membrane. So plasma membrane is selectively permeable or differentially permeable. Next is cell to cell recognition. अच्छा मु कहबी जनकर हाथ कटिगला नाही तार पोर्शन आर्मटा नाही आउ जने कौन करछि ना डेथ बेड रे अछि ही इज ऑलमोस्ट डेड मेंटली डेड ओके ब्रेन डेड के चाहूचंती जे ता आर्मटा नहीं कि से जो लोकर हाथ कटि जाय ताठे बर्तन कौन करा जाऊ लगे दिया जाऊ एटा जे डोनर आउ रिसेप्टर दे आर एग्री किंतु जेते आनी लगा जाउछि यार टिश्यू तार टिश्यू मैच करूनी दी जन नबा वाला दबा वाला राजी किंतु टिश्यू टिश्यू लेवल भितरे मैच करू नाही ताने किए कहूची ए जो रिसीभर जे जहाँ देहर लगा जाउछि आ जहाँ देहरु अणा जाउछि से दिहकर टिश्यू मिसमैच होछि ताले जो मिसमैचटा कहा लेवल रहउछि हु रिकॉग्नाइजेस दैट इट इज of my type or of foreign type na it is cell to cell recognition ए प्लाज्मा मेम्ब्रेन रे थिबा ग्लाइकोप्रोटीन माने जो बाहने बाहर फेस रे अछंती से माने कोउटा निजर कोउटा परर बा बाहर cell to cell recognition is carried out by the glycoproteins present in the plasma membrane. And next one is energy transduction. Energy transduction कहला माने फोटोन एनर्जी की एटीपी एनर्जी आउ कार्बोहाइड्रेट एनर्जी रे कन्वर्ट करजी हरित कणिका क्लोरोप्लास्ट. सो ग्रानम ग्राना जो पढ़िबा आमे ग्राना इज एनर्जी ट्रांसड्यूसिंग इट कन्वर्ट्स वन फॉर्म ऑफ एनर्जी टू अनदर. नेक्स्ट, so that is because of membrane. Without membrane energy transduction is not possible. Receptor function, for example आमे पढ़ुचंती बीटा सेल्स ऑफ पैनक्रियास तैयारी करछि कोन ना इंसुलिन काम दउछि कोठी से जाउछि ब्लड रे घुरी घुरी जाउछि किंतु से लॉज करछि कोठी जाई कि ना लिवर सेल्स रे आ स्केलेटल मसल सेल्स रे. स्केलेटल मसल सेल्स, सो स्केलेटल मसल सेल्स रे ए जो इंसुलिन व्हिच इज ए प्रोटीन हार्मोन ताकू किछि प्रोटीन अछंती से माने सी जाई कि सेठे लागुछि एवं सी ताकू चिन्हुछि जे यस दिस इज insulin, so that it is working. So receptor proteins are there in the plasma membrane. Similarly आमे देखुछंती जे कोरोना वायरस it especially affects the respiratory tract, not in the skin. Why? Because the receptors of coronavirus spikes are present in the plasma membrane of the respiratory tract, not elsewhere. Okay, so next, cell to cell adhesion. मु जदि एठो चिंता करी एटा तो मल्टीसेलुलर किंतु गोटे सेल को भिड़िले से कहीं बाहर की पड़ूनी ताले सेल टू सेल अठा दिया हला भलिया जॉइंट हेक अछंती, दे आर टाइटली पैक्ड, देयर इज adhesion, सेल टू सेल adhesion. आउ आमे अठा को इंग्लिश रे कहंती adhesive. तो मु एठे लिखि सेल टू सेल adhesion ताने हाउ दे आर केमिकली कनेक्टेड adhere to each other. ना इट इज द प्रोटीन्स ताने गोटे सेल और प्लाज्मा मेम्ब्रेन आउ गोटे सेल और प्लाज्मा मेम्ब्रेन भितरे जो कनेक्शन adhesion is possible because of the proteins. Next is membrane transport. So fortunately membrane transport is possible because of channels, carriers and pumps. As I have told you, most functions of the membrane are carried out by the proteins. Here you can see these are different pictures showing different types of membrane transport. एठे हमें देखिबा ऑक्सीजन. ऑक्सीजन कैन पास थ्रू, ऑक्सीजन टा होची गोटे न्यूट्रल मॉलिक्यूल, इट कैन पास थ्रू द हाइड्रोफोबिक कोर. हाइड्रोफोबिक कोर, कार्बन डाइऑक्साइड कैन पास थ्रू दिस हाइड्रोफोबिक कोर. ओके, they are small and non-polar. But if you come to glucose it is a big molecule, it cannot cross through the membrane. This phospholipid bilayer it is not possible, only it is assisted by certain proteins. से माने कोन करछनी एटा सेल बाहर जदी हैला आ एटा सेल भीतर हैला ताले they bind to certain proteins at this side, then the protein changes its structure and it is internalized. So आकु कहंती carrier protein which carries large molecules like glucose from one side of the membrane to the other side, they are called as carrier proteins. And channel proteins they are another form of proteins. They have a pore in the center. They have a pore in the center. एमाने आंकर structure change करंती ये protein माने. But these protein they do not change their structure. They have pore in the center and they have a door here. Once this door opens, gate opens, then the chloride ions, may it be, or potassium ions, they pass in large numbers from one side of the membrane to the other side, and this is another protein, okay, namely aquaporine. Aqua means water, porine means porine is a protein. So, this protein also facilitates through its channels the water molecules to pass from one side of the membrane to the other, because water is a polar structure. So, this diagram explains how molecules move across the membrane. Now, but when we see there is an infection, there is a virus or bacterium that enters our body, what happens to that bacterium? What happens to that bacterium? आमे जदि जूलॉजी रे पढ़िथिबा लिवर रे कुफर सेल्स अछन, कुफर सेल्स माने कोन करंती ना से माने जो बैक्टीरिया पस्थे आम देह को से माने तो खाई जानती. हाउ दे ईट अपन ना दे ईट अपन बाय एंडोसाइटोसिस. से माने ताकू इंटरनलाइज करंती तापर डाइजेस्ट कर जंती. आमे तो निहाती स्कूल लेवल रू वी मस्ट बी एक्वेंटेड विथ द टर्म amoeba. Amoeba is a large unicellular organism with cilia in its surface. Sorry, it is smooth, but paramecium having cilia in its surface. What they eat? Now they eat bacteria and yeast and small unicellular organisms. How they eat? We will see, and that is possible because of endocytosis. So, endocytosis and exocytosis are two cellular movements, two types of cellular movements, because we will see bacteria does not cross the membrane, it is internalized, but it does not cross the membrane as glucose did. So, they have different processes. So, one is exocytosis, okay? That means when certain things move out of the cell by membrane transport, cellular movement, that is your exocytosis, and another is endocytosis. मु ये एक्सोसाइटोसिस विषय कहिबे आमे समस्ते खाई के आसुछंती बा खाइले किछि समय पर्यंत लाळ बाहरछ कोठो ना सालिवारी ग्लैंड रो. ग्लैंड कंसिस्ट ऑफ सेल्स. सो ग्लैंड रो सेल्स भितर लाळ तयारी होची एवं से छोटो बेलून भलिया वेसिकल्स रे सब पैकेज हेक आसी प्लाज्मा मेम्ब्रेन सहित फ्यूज कर जाउची. सो बेटर टू से द वेसिकल्स पोर, ए वेसिकल माने आसंती प्लाज्मा मेम्ब्रेन सहित फ्यूज कर ढाली दियंती. दे पोर देयर secretion, they pour their content to the duct, exocrine glands, the salivary gland. Okay. So, exocytosis is an important means of delivering glycoproteins to the cell wall regions. Now, you can see this here. This is a vesicle carrying the secretory substances. This is the first step, this is the second step, this is third step. You see, this goes and fuses here. This is membrane and this is membrane. This is phospholipid, this is phospholipid. But this goes, एटा कोठु आसी थाए ना गोल्गी रु आसी थाए हम पढ़िबा. It is Golgi derived vesicle. In second step, it goes and fuse with the membrane, plasma membrane. Then all its contents are released to outside. So, this is exocytosis. In the same way, लालुआ substance आम सेल मानक जो सालिवारी ग्लैंड रो सेल्स मानक भितरे तयारी होंती, तयारी की वेसिकल फॉर्म आसी गोटे डक्ट को ढाली दियंती. सो दैट आमे देखिया जीभ तळे से डक्ट रो ओपनिंग अछि सेटा आसी बाहरे. ओके. सो एक्सोसाइटोसिस इज ए मेकानिज्म, बट हियर यू कैन मार्क एटा हस द भीतर साइड माने एई मेम्ब्रेन रो दिस इज आउटर फेस, दिस इज इनर फेस. किंतु से जते बेले मेम्ब्रेन सहित फ्यूज करछि इट बिकम्स इनसाइड आउट. ए जोटा भितर थिला वर्तमान इज तो मिसी जाउछि आ संग्रासी इनसाइड टा आउट हे जाउछि. दिस यू कैन आई थिंक इफ यू पुट योर ब्रेन यू कैन अंडरस्टैंड व्हाट आई वांट टू से. ओके. देन हियर एंडोसाइटोसिस. cytosis. Start करें. Endocytosis. Endo means within. Cytosis means into the cell. So it is the reverse of exocytosis. Exocytosis re substances, particles move from within the cell to outside, but it is reverse. One thing that is outer, it is present in the cell outer, it is internalized. You see the steps, this is the bacterium, it is the food particle. How this pseudopodia formation takes place, and pseudopodia formation sequentially internalizes this and makes a vacuole, and this is called food vacuole. Now if you see तामाने ये जो बैक्टेरिया टा बाहर थिला बा फूड पार्टिकल बाहर थिला ता चारिपटे मेम्ब्रेन एनसर्कल करी ताकू केमती गोटे वैक्यूल बनाऊँगी आ से वैक्यूलटा आसिला. बर्तमान यार भाग्य कौन ना आमे जंते खाद्य खाईदेले एंजाइम सहित मिसी जाए एवं डाइजेशन हुए. ये जो फैगोसाइटोसिस बा सेल इटिंग प्रक्रिया रे बाय द प्रोसेस ऑफ सेल इटिंग दिस फूड फॉर द अमोइबा इज इंटरनलाइज्ड. ये वर्तमान कौन हम ना या भितर आउ लाइसोसोम अच्छी लाइसोसोम डाइजेस्टिव एंजाइम अच्छी लाइसोसोम्स फ्यूज विथ दिस फॉर्मिंग द डाइजेस्टिव वैक्यूल. ताल फूड वैक्यूल एंड एंजाइम्स इन द लाइसोसोम जोटा ये देखा जाए ना दीटा जाक मिसिला परे एटा डाइजेशन हम एवं से जो डाइजेस्टेड फूड सब्सटेंसेस विल बी अवेलेबल टू द सेल बाय मीन्स ऑफ डिफ्यूजन. ओके, सो दिस इज हाउ internalization takes place. एठे देखिबो ए व्हाइट ब्लड सेल engulfs ए डिजीज कॉजिंग फंगल सेल. पूरा दिस इज स्कैनिंग इलेक्ट्रॉन माइक्रोग्राफ. तमाने एटा गोटे व्हाइट ब्लड सेल भूत भलिया लागुची देखिला भलिया आमे चित्र कर स्मूथली करी केमती, किंतु द शेप ऑफ दिस व्हाइट ब्लड सेल्स और मैक्रोफेजेस आर वेरी डिफरेंट. से नाउ दिस इज internalizing by means of endocytosis a fungal cell or fungi which causing disease. Okay, so से ताको खाई जाउची. सेथि पई they are part of our defense system के macrophages. Okay, so phagocytosis and pinocytosis. Okay, cell drinking and cell eating. Sorry, phagocytosis cell eating, pinocytosis is cell drinking. आमे जदि देखा ह्यूमन सेल रे जो egg acquires all its nutrients by means of acquiring liquid nutrients. So that is by means of pinocytosis. So this is all about what I intend to tell you regarding the structure of plasma membrane and function. Next we are coming to one important organelle, that is our nucleus. So nucleus, we have heard it in case of atom chemistry. Nucleus is center to the atom around which the electrons revolve. Similarly, nucleus is central to eukaryotic cell. So nucleus is the organelle, a very prominent organelle that defines the eukaryotic cell. सेथिपे इथे लिखा है that defines eukaryotes and differentiate them from prokaryotes. यदि prokaryote और eukaryote दीटा जाक टर्म यूज होची, eukaryotes have a true nucleus and the diameter of that ranges from 5 to 15. एटा mm हो जाइए, that is micrometer, millimeter ने, that is micrometer. So the size of nucleus varies from 5 to 15 micrometer. So nuclei are seen in the interphase cell of cell cycle. What I mean to say, आमे सेल पढ़ी सम विभाजन जो समय विभाजन हुए से समय सेल रे न्यूक्लियस न थाए, केवल इंटरफेज समय न्यूक्लियस एक्जिस्ट करे. दैट्स व्हाई द स्ट्रक्चर ऑफ इंटरफेज न्यूक्लियस इज डिस्क्राइब्ड. आउ जेमिति कहली एटम न्यूक्लियस इज टू एटम, सिमिलरली nucleus is to cell. If you see, this is a computer generated cell and this is the nucleus and this is other organelles. Similarly एटा को मैग्निफाई करा है, this is nucleus, यार 1/4 को काटी की बाहर कर दिया जाइए to show the different structures of it. Here I can show you. आमे जतो न्यूक्लियस पढ़ी छंती स्कूल लेवल रु डेफिनेटली आमे जानी छंती nucleus is a structure surrounded by membrane and nucleus at least contains the genetic material. and we know genetic material is DNA. DNA associated with histone proteins to form thread like structures called chromatin. You can see these coffee color threads tangled here, these are the chromatids, these are the chromatin threads, chromatin jalika jahaku ame padhichhanti odia re, because we have come from 10th to the 11th class and hardly we are acquainted with the English terms. So here I can say this nucleus is nasty. So nucleus ta bhitare nucleus bhitare gote little nucleus thanti thai, that is called ninyasti ba nucleolus. So this whole structure is a nucleus and this is little nucleus or nucleolus, and this is your chromatin threads. Where they float? The chromatin threads float, okay, in a matrix, liquid phase, fluid phase, that is called your karyolymph or nucleoplasm. जेमिति आमे पढुचंदी protoplasm, cytoplasm. Similarly, contents of the nucleus is your nucleoplasm. In the nucleoplasm, the liquid substance is there, the chromatin threads are floating, the nucleolus is there, and the membrane itself. Now, this is a computer generated model and this is a scanning electron microscope. Lekha is a nucleus of yeast cell. From yeast cell, this nucleus is taken and through electron microscope, ame tike terms न जानिले it will be difficult to understand. आमर ब्रेन टा अच्छी इंटैक्ट किंतु ब्रेन को स्कान कर देवा द्वारा ब्रेन रे कौन कौन अच्छी सरफेस रे पूरा जाना पड़ जाउछि. सिमिलरली न्यूक्लियस भितर कौन अच्छी जाना पड़ुनी किंतु न्यूक्लियस टा को जब स्कान करा हिछि ता सरफेस टा जाना पड़ुछि एंड यू कैन सी दीज आर द पोर्स. एटा को एठी हळदिया रे लगे कि ताकू मैग्निफाई करिछन्ती बट दीज येलो स्ट्रक्चर्स इन रियलिटी आर नॉट प्रेजेंट जस्ट टू impress ourselves. Scientists have made like this, but these are the natural structures. So this is a nucleus. It is a spherical, almost a spherical structure. I told many things. So ये जो बाहर structure टा, this is nuclear envelope. Where the nucleus lies? Nucleus lies in the cell. Question arises: is the nucleus is in communicating with the cytoplasm or it is isolated? No, nucleus must communicate with the cytoplasm. There must be exchange of things in between, because it is the organelle. It is the organelle in which the genetic material DNA is present. आमे आम घर गोटे कंप्यूटर रखिथिबा, इफ सेथुर आमे किछि एक्सेस करि पारनथि, पढि पारनथि, प्रिंट आउट नै पारन, तार किछि मीनिंग नै. Similarly, nucleus having genetic material must express, it must produce proteins to form or to express itself. Now, this is the photograph that we have taken in our laboratory. This you people can take in the first year class. This is onion fleshy leaf. आमे जो पियाज खाउछन्ती, पियाज को खाइला समय देखियो भांगिला बेले गोटे जरी भळा लेयर बाहर जाए. आमे किछि करि नाहु, आमे सेल जरी भळा लेयर बा एपिडर्मिस टा को नेइचो माइक्रोस्कोप तळे थोइ दिछु. And these are the cells, and these are the nuclei. तमे देखिबो एठे न्यूक्लियस एठे सेंट्रल अछि आ कोठी पेरिफेरी अछि. नो, it is always in the periphery. Time doesn't permit, but I can say in the plant cell nucleus never lies in the center, because plant cell ro मझी portion ta majorly occupied by रसदानी, tonoplast. That's why nucleus is pushed to one side, कांथ side की पड़ी थाए. Anyway, a nucleus is delimited by a nuclear envelope. Here I can show you. This is the nuclear envelope. The nuclear envelope consists of two membranes, outer membrane and inner membrane, and they are separated by a perinuclear space. But nucleus, as I told you, it is not an isolated box, rather it is punctured by many pores. तमु लागुची ए जो सेगमेंटा is separate from this, अलगा अलगा, किंतु नुहे. मु तुमको गोटे एग्जांपल देवी. आमे गोटे नड़िया रो सढ़ईटा नबा, पूरा गोटा सढ़ई. ओके, आमे जदि 10टा जगह तक कणा करि दियंती सेटा कौन अलग अलग हो ना सेमती थी विथ होल्स सिमिलर इज द केस इन केस ऑफ न्यूक्लियस इट इज punctured by many pores it is these are not simple pores these are guarded by proteins we have door is an opening but it is not open फुंगुड़ा नहीं rather it is guarded by two panels constituting the door similarly these are the pores they are guarded by the proteins so सेति पय आकु कहा जाउची न्यूक्लियर पोर कॉम्प्लेक्स जदि हम पचारिबा in a plant cell nucleus how many pore complexes are there it is almost 3000 pore complexes are there in the nucleus okay आउ गोटे जिनस मार्क करियो गोटे ग्रीन कलर लेयर अछि ता भीतर को एठी that is called nuclear lamina means layer that is formed of the protein fibers that makes the structure spherical so the spherical shape of the nucleus is maintained because of the nuclear lamina present just below the envelope and these are the chromatin network and this is the nucleolus. Next, so here I want to emphasize this is the outer membrane, this is the inner membrane and both of them are continuous at the margin of the pores, they are not separated. Remember, outer membrane it is a phospholipid bilayer, inner membrane it is a phospholipid bilayer and एठी जो टिके नागु देखा जाउ that is the perinuclear space okay that is about 40 nanometer thick a space compartment. Now this outer membrane is continuous with the endoplasmic reticulum as we will see and as it is a computer generated model you can see the pore complexes. So this is the original photograph through scanning electron microscope and these are further enlarged and this is the computer generated model. I told you 3000 pore complexes are present in the animal cell and so many number in the plant cell. Okay. So now inside the nucleus there is nucleoplasm in which chromatin threads comprising DNA and protein complexes are there, enzymes are there and many RNA polymerases because nucleus is the site where RNA synthesis takes place. The enzyme for RNA synthesis is your RNA polymerase and proteins are present. Histone proteins are also present therein. Water and salts, many substances are present in the nucleoplasm. Okay. And here at least it must be noted that chromatin, we have read of term chromosome and chromatin. Chromosome is the distinct bodies, they appear during the mitosis. विभाजन stage रे मध्यावस्था जो मेटाफेज समय chromosomes are distinct, but in interphase stage they exist as chromatins, the colored threads, they are nothing. DNA molecules complexed with histone proteins form chromatin. So in this stage they carry out gene expression and replication. Nucleolus जहाँ को हमें पढ़ुचंती निन्यष्टि, this nucleolus is the site of rRNA synthesis and ribosome synthesis. We will come later, but let me tell you ribosome has two units. Bacteria case रे 50s and 30s subunits are there. से माने कोठे तैयारी होंती ना, they are synthesized within the nucleolus. So nucleolus is the site of rRNA synthesis. Proteins come from the cytoplasm, there they assemble and ultimately ribosome subunits are assembled therein. So this is the function of nucleus as a whole. Nucleus is the genetic repository of the cell where chromosomes containing genetic material are stored safely. So this is a distinct compartment carrying almost the genetic material of the cell. So it is the house where the genetic material is stored. Second is it is the master organ of a cell. It controls all the genetic and metabolic activities of the cell. That's why it is called master organelle of the cell. Nucleolus, the little nucleus, ninnesti, being present on the nucleus, synthesizes all the ribosomes required by the cell. So ribosome synthesis that is taking place in the nucleolus and subsequently they are transported to the cytoplasm where they work through the nuclear pore complexes. Similarly, mRNA is also synthesized in the nucleus and they are transported to the cytoplasm for protein synthesis. I think in the two major organelles, one is plasma membrane, I spoke a lot, and the nucleus, I think you people must have understood this and try to go through these slides to make you understand better. Thank you all.